to your wives, husbands, families, jobs, whatever. That was not the intent, and I'll try to make it better second time through it. And thanks to the few people who came up and gave me analysis, I felt like a quarterback who just lost a Sunday game. Well, you really should have done this. Okay, I appreciate that, I did. So, you know, that's how it goes. But anyway, it wasn't out of disrespect, it was out of, well, actually trying to do the right thing, but not really getting there on my part. But I will say, I was very impressed that usually some people will quit exams because they just give up. The fact that people are still writing for such a long time meant that you found something to write about and you felt you had something that you could contribute, which I'm looking forward to seeing what that is. I will also tell you I have not gotten to the exams. It is exam time. I'm either writing them or correcting them as we go, but I'll try to have those back to you maybe Thursday, definitely by Tuesday. Okay, put that behind us for whatever it is. If you're mad, well, come and yell at me. If you're happy, come and laugh with me, whatever. We'll try to get through it as a class. I wanted just to take a quick look at your responses for your reports, and let's just see what people have written down. You can probably have looked through these yourself, but we want to make sure no one's writing in anything else. We also, remember, when you're writing genus and species, capital on the genus, small on the species, and italicized. If you didn't get it, write it for the report, please. I realize you're just writing answers here, but so we got chlamydia, that's always a popular one. Bacillus, take a look at that. That might be on the list of things that are on the blacklist. Lactobacillus, rhamnus, vorticella. Is that a protozoan?
Yes.
You need a species with that also.
Okay.
Pick one. I mean, even if you stick to the genus,
Yeah.
do the species for the sake, because it's a species report. Thanks. I like Deinococcus. Hard to, Deinococcus takes more radiation than any other known thing. Why? Alex, probably tell us. Clostridium difficile, yes, big problem. Clostridium botulism. Klebsiella, might be on the list. Check on that. If you still really want to do that, let's just talk. If that's just like your passion to write about Klebsiella or Bacillus subtilis, I just don't want to get a lot of overlap. Neisseria gonorrhea, always a popular one and worthwhile. Tobacco, good. Meniscipium tardigrades. Tardigrades? Cool. Treponema, oh yes. Clostridium, I'm changing. Oh wait, here's mine and I'm changing. Okay, tuberculosis. Oh well, should be hard to keep that one short, but that's a Plasmodium, Lactobacillus, rabies. Okay. Pseudomonas, check on the blackout list for that. Oh, just saw that. Oh, okay, good. Okay, so you fixed it. Great. All right. Yersinia. Leuconostoc, yeah. Okay, well, as a group, Porteous, check on the list on that one. Myco and leprosy. Okay, looks like we have a good amount there. I'm so busy I didn't have a chance to look at, but if you need to revise. If you're still passionate about one, let's just talk on that, just so we have minimum overlap on what you're covering. I think that's the big deal. So I think that'll work out. Okay. So we left off with viruses. We know their size. I'm sure I have an idea of the size. A little bit of how they replicate, and they don't really reproduce. And you may think that's semantics, but at least there's something behind the logic on thinking about that. And we're talking about shapes and their genomes, and we'll return to the genomes a little bit more today when we get into the Baltimore classification, which is a bit lot in the beginning, a lot to take in, but as we tackle the groups of viruses, you'll see that Baltimore had a really good idea on how to do it, so we'll come up to that. And I believe where we left things off is not only do the viruses replicate, but that they do so in in an orderly fashion, which is kind of interesting to us, because remember, they're not really dealing with operons or promoters and all that kind of stuff. A lot of it has to do with probability, when something gets incorporated and when it's expressed. So, you know, you gotta realize that they're coming in with a really, really small genome. You know, if you compare it to what's going on with the bacteria and other cells that have metabolism. And this was up the other day, and of course in your notes, but we're looking at a time scale, okay? Always look at what you have as being presented to you. Time, and that's giving us the relationship with the relative virus count. So they're looking at really the assembly of the virus, the virion, as opposed to the prophage, the stage of the virus that's incorporated into the host. And so I think we've been through it, but it's worthwhile looking at it again. I like this timing thing. So the virus is added, in this case, we can just think of it as a culture and a phage. That works pretty good for us. And then we have the virus is added, the early enzymes are expressed. These enzymes usually have to do with replication of the genome, so they're polymerases. We have nucleic acid production. The later genes that are expressed are the protein codes. So the reason this is flatlined and so low, this could essentially be zero, because you know, I mean, there'd be a big peak here. You added viruses and then they went away into the cell, so that's kind of what's happening there as we pick up the story. And so we don't really have any virus particles until they assemble right here in the maturation stage. At that point, as they begin to assemble, this number goes up, and you know, in the cell somewhere in here, they've assembled and released due to burst size or the expression of an enzyme or other factors, and the viruses, mature viruses, are left out. So this period, this latent period, is just when, you know, if that's in reference to the study of virology. That is, it's latent in that you cannot go in and count viruses at this point. They're incorporated, so it's a terminology thing. We'll talk about latency also when viruses crawl into like your nerve tissues and sit there and are expressed at different times, like herpes and some of those viruses. All right, so and I think this went with it. Early proteins, what we need to replicate nucleic acids, so the polymerases are usually the ones there. I'm a little bit vague because sometimes we have an RNA genome and a DNA genome. So I mean, there's different enzymes at hand there. We're being general here. Late proteins include the virus coat. Could also include the things like if you had T4, it might be the tail and the fibers that go into that also, depends on what virus you're talking about. And also some of the spikes sometimes are expressed in there also, depends on the virus. Okay, so here's T4 going in and essentially just showing you what's going on. I won't play it for you. There's about a, I don't know, 30-second, 45-second video where an artist has done this very nice rendition of this, but basically we see what's going on. We have the T4 coming in, finding usually an area of reception to mount and position itself. And then actually, this is about right. It uses part of the tail, as opposed to the tail fibers, above the plate to inject, in this case, all the way through the outer membrane. This is shown, of course, as being, you know, a hugely wide thing because, you know, we're dealing with a cartoon, but, you know, it's not all that big. The other thing in here is that you have to have some way to get through the peptidoglycan, and so lysosome or maramidase, we talked about that being in your eye, that enzyme is fairly common. And then getting that genome finally to get through into the cytoplasm. Okay, T4, we're dealing with double-stranded DNA with these guys, so that has to be expressed into plus sense messenger. We'll talk about that a little bit more, remind you, but I know we've talked about it a little bit, and then we'll get into the expression of the genome and some of those things. So this is the cute little video. Tested it this morning. Seems to still work. Very nice. Take a look at it when you go over the slides if you haven't already. It's a nice animation of what we just saw on that slide. So, viral attachment penetration. Many bacteria employ restrictive modification systems to evade viral infection. Do you remember what restriction enzymes are? In maybe you learned in genetics or part of something else. Anyone, does that word restriction enzyme bring up anything in your memory? And we used to talk about in this class, but we moved so much of that stuff into supplemental. I can't remember if I covered it or not. Well, restriction enzymes exist in cells to look around, and just for that reason, to look for certain patterns and to cut out DNA that might be in suspect of being part of the genome, the actual organism's genome. And they have very specific types of nucleic acid sequence. You know, sometimes the cut is straight across both DNAs. Sometimes it's a sticky end cut, or it's a little bit uneven. But anyway, it cuts. And so some viruses go, over time, the viruses that have figured out, in a way, how to modify restriction enzyme sites, sometimes by just methylating or changing the site, can evade being cut out of the genome and can still infect. You got to realize, less so, well, it's still a selection game. And I have a slide on this because I want us to think about viruses and selection. It's easy on one hand to say they're not living, therefore they don't evolve, and they're not subject to those rules of natural selection. On the other hand, you have to look at natural selection many different ways, but one way is to look at it as a probability game. That is, who is around, of the things being selected for to continue moving on through different generations. And if you, have certain modifications over time that allow you, the virus, to continue to infect and replicate, then you're selected for, of course. So evolution, in a way, is a factor. Just kind of talked about, the modifications. I said methylation, there's also glycosylation. That's absolutely true also. For that, if that nothing comes to mind like a methyl group or a sugar group, you just write down the side. That's what I mean, is that the restriction enzyme puts on a very simple little modification, sugar or methyl group, and that the enzyme, as you know, is very specific, because, I can't get to that site anymore. If you take genetics, have any of you taken genetics? Do you do any restriction enzyme work? Do you remember cutting up DNA? Okay, I don't know if they do it in general biology anymore, but restriction enzymes we use in molecular biology to cut up DNA, and that was really important early on when we had to cut up DNA for sequencing and other things. So people use restriction enzymes. There's many of them that are very well defined. So it's no secret at all. But on the other hand, getting around the restriction enzyme is kind of this clever little trick that shows up here and there in the system. Now, David Baltimore gets the credit working with others. Most scientists do, but a lot of times you'll hear a certain name given to certain people. But he started to look at this world of viruses. Okay, 1975. Who's alive in 1975 in this room? Okay, I was pretty much alive. I was getting ready to graduate from high school. I was going to sleep at night wondering if I was going to get sent to Vietnam. So that's the kind of thing. They ended the war right about the time of my graduation. Thank goodness. So right around 75, but I remember growing up. So that's the generation I speak to you from. Okay, so anyway, in this time period, craziness all over the place, these guys are sitting around going, okay, viruses, big deal. We get that they wipe out people all the time. They wipe out plants, they wipe out everything, and there's some neat things about them. Let's take a look at them. What else is going on? How are they classifying viruses? EM was working, so they had electron micrographs. They saw different shapes, and they started classifying things by shape. realizing probably what you do, what you're saying. Well, I can tell a little bit about my bacteria. I'm starting to identify bacteria. I can tell a little bit by the shape, but, that's not really a reliable characteristic. When it comes down to strains, comes down to antibiotic resistance, all those things. Okay, shape helps me, but that's not the only thing. So these guys said, well, let's look at viruses and let's look at this DNA. Remember, DNA now and RNA is in the cards. I mean, only around for about 20 years now since we know about DNA, but people rapidly figured out that. Okay, it sounds like an eon ago, but there's still some of us who are alive and remember that time. I was not thinking about DNA in high school, I'll tell you that much. Anyway, so the key here will be mRNA. Okay, so that's just very easy review. The process of DNA going into messenger is called what? Just so we're all together on this. Transcription, and then if we're moving messenger into protein, that's translation. Okay, transcription is kind of recopying the message. Translating is translating it from one message to another, nucleic acid to amino acid. Okay, that's going to be important to us. And I'm sorry, because I'm from that generation, I forget a lot. Have we gone over what negative and positive sense RNA is? Sort of. Okay. Well, let me go over it again since I sort of did it, and if there's any sort of fuzziness, let's work that out, because that's going to be important in the Baltimore classification. Okay. So, when we usually learn the molecular dogma, we learn, okay, there's DNA replication, that's important, but then we learn DNA into RNA, and they always just show you one part of the DNA molecule, the template that's being transcribed, and then you just take that into amino acid sequence, right? You don't spend a lot of time, I don't know, a lot of classes don't spend a lot of time saying which side's the 5' and the 3' and which one is the sense and antisense strand. Okay, so this becomes important here. So from the DNA, if you have two strands of DNA, basically two strands of a gene, one strand is being transcribed. That part that's being transcribed, that's the template strand. Okay, that strand produces, in the way we've typically learned it, mRNA. That's your plus strand mRNA, because that goes straight into being able to be coded for the amino acid. So as we've learned it, we're just putting out labels on the way we typically learn this in many classes. And I don't know, maybe your class is a little different. That's the way I kind of learned it. Okay, now we realize, let's just move back to DNA again. So one strand went to the mRNA, went to amino acid, so we're giving that plus. Okay. The other strand, okay, if we were to transcribe that, would give us a negative sense mRNA. It'd be something that, you wouldn't have the start codon, the AUG in the right spot. The stop codon would be reversed, and so that it wouldn't make a lot of sense to the cell, plus amino acids would all be coming out different from the degenerate code, right? Okay. At this point, who's lost with the words I'm saying? Are you hearing words, or are you hearing blah, 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 code, blah, 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 plus? Okay, and can I help you before we move on? Everyone? Okay. I mean, not a problem.
I'm following.
There you go. Okay.
Quick
Good.
question.
Yeah.
So the double strand of DNA is the one that runs just canonically from the 5' to the 3'. And the other strand is the
Would
one
be the
that
3'
produces,
the semi-conservative or the anti-parallel.
the one that produces the oxidizing fragments is the one that produces the negative mRNA.
Yes.
Okay.
Some genes, though, can go the other way, and they can flip and do the other strand. That's why they go with plus or minus. So it's not always the same. But think of a gene,
Okay.
and then stick with this one. But yeah, you see those genomic maps, and sometimes the arrows are this way and they're that way, because it depends upon which strand is being looked at.
Okay.
That's why they go plus or minus, because the primes the 5' and 3' can't always, you can't say, oh, it's the outer circle, and you always use that, you know what I'm saying?
Yeah.
You with us? Okay, no one's saying no, so I'm saying, thinking yes. If you're not, it's okay. I was too embarrassed to ask a question in class. Come and talk to me about this. It's an important fact. And we'll go over it again, diagrams, shadow puppets, whatever we need to get the job done. Okay, now, we got to the sense. Baltimore says, let's classify viruses, okay, by how they get to the plus mRNA sense molecule. That's it. So when we look at his classifications, their classifications, it'll always be, okay, what are they doing to get there? Okay, and I'm pretty sure I got this queued up, right? So let's go new copy. Okay, so here's the big thing: positive strand, single strand mRNA genome with the same orientation as its mRNA. That's basically saying what we said, that it's ready, it's messenger ready to be translated. Okay, negative sense virus, single strand genome. The complementary orientation doesn't usually make sense because codons and stuff are in the wrong spot. Okay, retroviruses, I will come back to them. They're kind of like, huh, what's going on? They go against the molecular dogma, right? Okay, they're the ones that basically go against the molecular dogma. They have RNA, they go into DNA, then they go through the whole thing because that's the enzyme they have. Okay, but it makes for a really interesting little evolutionary turn in the stuff. So, okay, so formation of mRNA and new genomes and DNA viruses. So here's our double-stranded DNA. Okay, so if we have double-stranded DNA, which has a plus and minus strand, okay, because it's anti-parallel. Okay, one is going 5' to 3', the other one's going 3', 5'. Okay, in there you can you can basically transcribe mRNA because the copy for the plus strand is in there, and then you can go ahead and do this. So DNA, double-strand viruses, DNA viruses, DNA plus viruses, class 2, can synthesize the complement and then also make M-strand RNA. Okay, now RNA. RNA is interesting. So if the genome of the virus has the plus strand and the minus strand, okay, it can make the plus strand from that. Okay, so that has to happen. Transcription of the minus strand. In the single strand RNA, that's messenger that can be used right away. And my question always is in virology, and I'm just not, I don't know a virologist to ask this question, is why isn't there selection for everything to be like that? It just seems so fast and so efficient. I do not know the answer. Perhaps some of you will find out. I don't know. Okay, transcription of the minus strand. Okay, so you can make plus strand. Okay, if you have the plus strand RNA, you can make the double strand intermediate if you have reverse transcriptase and get that over there. So they start making these different classes. I can tell you right now, I won't ask you to memorize the classes. I might give the Baltimore classification again. I mean, I think if you use this from day to day, you probably learn the classes. And if in a class in general microbiology, can you use the chart? Can you explain what's going on? Then I think that's what reference tables are all about. So this is basically the same kind of idea in diagrammatic form, just talking about if you have plus RNA, you can make the negative DNA, that can be double-stranded, get that off, and you can make the mRNA. It's just the different avenues. With your plus RNA, you go straight into mRNA. Either way that you look at, they summarize the classifications. So, all right. So when we look at these, the main thing is for the Baltimore classification is to look at how you get to the plus messenger RNA. That's the case. When you see, just to decode things, there's a lot of viruses out there, just like there's tons of bacteria out there. So what you want to see is if you see these different classes, you know, you could look that up, okay? So it's not a mystery to you. It's just like in decoding the Burgess manual. There's terms in there that I hope I can help you to at least go, okay, I know what that means, so I can look up what's going on. Here they're just giving you some examples. Double-strand DNA one. This one we already have gotten examples through. So T4 is pretty much kind of our, we love E. coli to talk about E. coli. We love talking about T4 because it's got that funny little lunar lander thing. But there are not to get mixed up, there are separate viruses that can account for some of the pox and herpes virus, single-stranded. The FEX174, we'll talk a little bit about that in this class, just because it does circular replication, but that does show up where we have your polio, the single-stranded RNA. Our favorite retrovirus is HIV. Not favorite that it's a favorite that it's in the news a lot. That's what I mean by that. It's favorite at all. It is what it is. It's a terrible disease. Okay, I think that ends this, and I want to go into the next part swiftly and get through viruses today if we can. Okay, so we're going to talk about viral diversity and start marching through. Now, up until this point, and I can put these out there again. Someone wrote to me over the weekend and said, I'm confused what pages, and I was going to put it on Blackboard. I'll try to get up, but if you're reading through the chapter, really that part into the Baltimore classification, pretty much part of the chapter, I don't have my book with me, it's the full suite of pages, and I'll look it up. But the next ones, when we go through these different diseases, just pick those sections when you're reading through it. So when we go through rabies, go look at rabies. I mean, not that reading about all the other viruses is not cool. It's just like reading the whole book would be a wonderful thing for everyone to do. But there's just, there's hundreds and thousands of millions of bacteria and a lot of viruses. So we're going to pick and choose a few more of the popular ones, talk a little bit about them, and read those sections. And we're running around like crazy today trying to get stuff ready for the unknown test in 346. But if someone reminds me, I'll get those pages up for you. I was in the car when I was trying to get those pages to someone, so I didn't write them down. Okay, if you could come to the lab now, there's a picture in the book, I think it does show this now. You could come to the lab and you could do this. Okay, we have this stain, okay, which is called YoPro-1. I don't know the names, that's it. Okay. Jed Furman, alive, kicking, wonderful, amazing microbial ecologist, primarily in marine systems, but if you want to read someone who does wonderful work, Jed Furman's there, and he offered this picture. This is what I saw when I looked through the microscope and I stained a sample of water, much like many of us in labs stain a sample of water with DAPI. And when we looked through it, we saw these bigger particles. These particles, our particles are nice little dots, sometimes a few little rods, but there's big blue particles. And we all went, this is so cool, Dr. Lemke. Can we do more of this? I said, yes, come to my lab and we'll all do more of this. Anyway, that was DAPI. So how did DAPI work? If you're not in the lab, what DAPI is a nucleic acid stain. It goes to double-stranded DNA, which bacteria have if they're alive, and if they're dormant. If they're dead, they may or may not, depends upon if they degrade or not. So it's kind of a confusing picture, but stains are like that. Stains, sometimes stain what you want, but they sometimes stain the paper alongside what you want. You gotta interpret it. Okay, so we saw this in blue. Were there double-stranded viruses in that sample? Oh yes, many thousands. Okay, I don't know, maybe millions, because we're talking about 1 billion viruses in the middle of water. Oh yes. Okay, why didn't we see them? Well, one, our class, and if you're in lab, come on, I'll show you this any day of the week. I love looking at these things. we're just learning. Maybe they're in behind. Every once in a while I can see blue specks and I'm not sure of them. Okay. But, and also there's a bunch of viruses out in nature that are not double-stranded DNA, and Dampy doesn't do a good job of doing that. YoPro does, it is a fluorochromic stain. You have to use a different excitation and emission filter to make it work. But it glows super, super, super bright when it's hit with the right wavelengths. And that's why we can see these viral particles. So when I called up Kurt Settle, I was amazed. You're probably going, oh yeah, it's just a bunch of little lights on it. I was like, I can't believe I'm seeing viruses, Kurt. He says, Lemke, you idiot. Yes, there you go. Lemke, you idiot. Which they'll probably chip on my little tombstone as they heave my body into the abyss. You know, Lemke, you idiot. You know, you're not seeing the viruses, you're seeing the glow from the viruses, which in retrospect he's a little bit wrong. I mean, the viruses are there someplace, but you cannot see. Okay, Kurt. One for you, I'm watching. Okay, anyway. But this is it. So you can tell, perhaps, of my amazement. You know, in this we can see a smattering of bacteria, and this would maybe be in 0.1 mil of water from any water sample in North America. Go look at the paper I wrote on this. It's amazing how constant bacteria are in nature. I don't know why. If you really think about this as an ecologist or evolutionist, it's a big question. But look at these viruses. Okay, this is not uncommon. They're amongst us all the time. What can we learn from this? Well, a question that's still open today. Oh my goodness. There are so many viruses out there. What are they doing? Are they infecting other bacteria? Yes. Are they taking out algae? Yes. Are they causing disease? Yes. Is there a whole bunch of them? We have no idea what they're doing? Oh yes. Okay. And that question is out there. You know, we don't really know the answer. Is it an artifact? Perhaps. You know, are we just making little pieces of clay glow? I don't know about that as much. These guys are pretty careful. When we did this before, you used to have to do the slide, let it sit for like a day or two in the dark, and then do it. Now there's even a microwave version, like so many other foods in the world, but so many other things. And they are looking at these much more quickly. You want to know my big question for it? Anyone care? Sure. No? No big. My big question is, why can't viruses be food? I've never seen a paper on that. Okay, you're going, huh? They're so small, but if you got these little things filtering out so many other things, why aren't they food? And I mentioned that to a few people. That's a great idea. You should look into it. Well, you know, in my spare time. But I've often wondered that because look, protein, nucleic acid, nitrogen, phosphorus. You know, oh boy, that seems like that'd be a food source for something that's starving in the environment. Even if you're small, a bunch of small stuff really adds up. Don't know. Okay, so what? I'm going to go past this. I forgot that this one was in here. That one explains the evolution of viruses, and it's complicated. Maybe we'll return to that, but I want to get into an overview of the different viruses. Evolution of viruses is tough. You'll never see them in the tree of life. They don't have ancestry because they don't reproduce, at least in the way that other things typically reproduce. Okay. Therefore, as you track virus lineage, you often have to track the host lineage, and this gets very, very complicated. I would like to go past that and get into talking about viral diversity, just for time. So, overview of bacterial viruses. So we know they're called phages, so that should be our bacteriophages, phage for short. Okay. And we know that the bacteria that are studied the most have the viruses that are studied the most also, and so E. coli and Salmonella are on the top of the list. Most of these are double-strand DNA. Most of them are naked and don't have the envelope. They just go straight for the jugular, bacterial jugular on this. Okay, and here's a smattering of some of these that we're looking at. Some are single-stranded RNA. The ones that are probably the most popular are these. We will take a very brief look at SS DNA, the FEX174, in a few moments, but this is probably the poster child of the phages, the T4. Okay, so in this case, we've talked about bacteria going through doing the lytic phase. They lyse or kill. Another rendition of how these things inject their DNA. One thing we will talk about in a few minutes here is the temperate phage. Okay, the temperate phage, the viral genome replicates with the genome of the host. Okay, so we've learned it that the steps where you have attachment, penetration, incorporation, maturation, and then release. In temperate mode, you'll start out, you'll have attachment, penetration, and then there's incorporation into the host. Okay, this is where things change. When you go into temperate phage type of infection, then the phage genome is incorporated with the host. So every time the host replicates, then the virus genome replicates also. And you go, so what? Well, it is kind of a so what moment because these viruses can sit in the background for generations of bacteria. Okay, but it's kind of a smart thing. So now you're in all these different other bacteria that are different than their parent bacteria generations, that they have virus DNA in them, and you're getting many copies of that. Okay, then what happens in this type of cycle is there's some kind of signal. Okay, sometimes, I mean, you'll read in the book there's UV. It's usually not, UV does trigger it, although there's not too much UV usually in the gut of people. So, but that's something they do in the lab. But there can also be a chemical signal that goes to the cell that stimulates the virus to go back into the lytic cycle, and it goes in, and it does its usual infection, assembly, and lysis. The neat thing is oftentimes when a virus has the prophage incorporated in the genome, it can't be infected again. So there's some kind of evolutionary benefit to the cell with that. So it's kind of a touch and go kind of a thing in that. Not much said there. I think we've covered all this. The T4, the lysosome, get through there. And we've also, I think, covered this, although I kept this slide in because I like the way that this lays out the genesis of the virus particles. Because before, where it was very general and it was showing us the viral particles, here there's really a lot of emphasis on this early phase, this early phase that then allows the DNA replication, which gives way to the other proteins that can be made in RNA and leads to the maturation. So this couples nicely with the slide we had in the beginning, but I put it in a T4 because that's where it is classified. These are just scanning. I think these are SEMs. They're shouldn't, scanning EMs. I think these were taken at SIU Medical. One of the reasons I was drawn to them, but I don't see it on there now. Either way, here they're showing you dividing E. coli cell, and then they just show you the burst in them. So in a way, so the thing to take away from here is the disruption of the cell. It basically blows the cell apart. The thing to realize that's artifact is that in SEM, there's probably not these big strings in here. What you're seeing, though, is something you know about. That's usually capsule or extracellular matrix because it's 95% hydrated. When you make SEM, you gotta get rid of all the water, so that big sugar coating turns into a stringy mass. People used to think bacteria were hung together on webs and strings. not a lot of people, but early biofilm scientists, they realized that it's an artifact. So, several things to note there. Okay, here is the summary of the lytic and lysogenic cycles, if you will. At this point, you should have a pretty good idea of this pathway, so I won't review that too much more, but you can see this is a nice, because it's got the visual, it goes along with my narrative attachment, okay, injection, incorporation. Now we have this incorporation, especially if these double-stranded DNA viruses, into it, so it's a prophage. Before it's a phage, it's just the genome. Okay. And that's in there. So every time the cells divide, you get this. And this shows that it kind of stops here, but you can have several generations and a big proliferation of the infected bacteria, okay? And then signal, can be various different signals, depends upon the cell, what's going on, but it'll induce it then to start replicating the viruses, so they come out of their little DNA, make more viruses, assemble, and then lyse. It's worthwhile because there are different evolutionary implications for if you're just going in, infecting, and destroying, okay, and then, as opposed to if you're going in, infecting, regenerating, okay, and then going into lysis, okay, as far as what goes on and being incorporated into the genome. In transduction, something you may or may not have, transduction happens with the temperate phage. Okay, there's three modes of moving around genetic information: conjugation, transduction, transformation. How many people know about conjugation in bacteria, not what goes on in paramecium? That's a different conjugation. Okay, in a nutshell, for these three things, we're going to talk about transduction, one of the viruses, and I'll get this into one of the lectures. I thought this was covered someplace else. Okay, conjugation is when a plasmid from a bacteria travels through a pili. Remember early on we talked about these pilus? Okay, if there's a conjugative pilus, okay, there's a little tube between two bacteria, and the plasmid unrolls and starts moving through the tube to the other bacteria. Okay, so sometimes you see this referred to as bacterial sex. Okay, no, it's not a little bacterial penis, and there's not a little nothing like that. Okay. They call it because of the movement of DNA between two organisms, somewhat like, I suppose, fertilization is thought of. Nothing's being fertilized, it's a movement of DNA. Okay, but sometimes the popular media talks about conjugation as bacterial sex, very similar as some people talk about weapons as being nature's kidneys. That's how people talk. Okay, so that's conjugation, that's movement of genetic information. The other one that we'll talk, I'll get some slides on this, is transformation. Okay, does anyone remember Frederick Griffith's famous experiment in the 40s or 50s? Have you heard of it? With the mouse, you've heard of that? Okay, I'll get some slides on it, but what Griffith did, and they didn't figure out for decades, is that he had two forms of bacteria that one would kill mice, the other one wouldn't. And if he heat killed the one that would kill the mice, it would not kill the mice. So then if you put the two types of bacteria together that should not kill mice, all of a sudden it would, because the dead bacteria's DNA would go into the live bacteria that would not kill mice and then kill them. And you go like, wow, now, I know it's confusing, I'll get a slide up for it, but Griffith's experiment with mice is a pretty big deal. And the big deal there is transformation. That is, DNA that's free can be picked up by cells. And at first point you go, oh, that's cool. And then the second, when you think about it a little more, holy smokes, that's kind of scary. DNA can just be picked up. Yeah, there's even a paper written, and the way that molecular biologists do it is by sometimes heat shocking, and sometimes through using electric voltage, electric current. Okay, it makes the cells leaky so they can pick up DNA. There's even a paper that talks about lightning strikes, that lightning strikes to the earth may increase transformation in bacteria. It was more conceptual, I don't know if people follow that up, but the fact that you can change expression of a cell by it picking up genes is a big deal for people. I'll put that in there. I thought that was background material, but we'll cover that a little bit more detail. Well, in transduction, okay, transduction is the other way of moving around genetic material that people classically talk about, aside from jumping genes in McClintock's work. So with that in transduction, if things go normally with the lysogenic phase, you'll have the cells incorporate the viruses. The viruses will finally be expressed. They all coat up and make the viruses, and the viruses go their way, and the cell dies, and that's the end of it. In transduction, you get some of the host cells' DNA caught up in some of the bacteria. Okay. So what happens is instead of just clipping out this part, making the virus, okay, it may clip out either part of the DNA from the host, or sometimes they just get most of it caught up in the genome. So now you have this virus, this infective particle that's going around and infecting other bacteria, okay, with another bacteria's DNA, and that can cause changes in the expression of other bacteria. That's called transduction. So as a review, I know it's a lot of words. Conjugation is movement of DNA between two cells that are close. Transformation is the picking up of free DNA, largely. And transduction is moving around DNA using a virus and the lysogenic cycle. All this is well documented, and I'll try to get some slides for you on that, but it seems like an opportune time to talk about that, and we'll review it. Maybe we can get to genomics next. Okay, let's go through a few more viruses then. We talked about the temperate phage, not much to look at, but some of the T phages will do that. In this bacteriophage, the T1, I just wanted to show you a mechanism that they use for their genome that we see also used in the replication of plasmids in bacteria. And this is called rolling circle replication. Most of the time, we learn replication as a strand of DNA, opens up, and then we have the leading and lagging strands being replicated, right? The Okazaki fragments and all that. This is taking a template of the double template and basically putting in the primers. It's either DNA or in nature RNA, where the polymerase can start, and then the template just keeps on rolling, and the replicate complement just keeps coming off like a machine. So it's kind of like a drum machine. Okay, so you just another copy, another copy, another copy. Okay, as opposed to the replication forks and that kind of thing. So the point here is when you have the circular DNA, rolling circle replication is a real fast way of making a lot of copies. Just rolling it off. Don't have to worry about replication forks and Okazaki fragments. Just as fast as you can roll it, it can replicate it. Just another way of showing it. You have the nicking enzymes. There's always a way of opening it up. Okay, and then you have the copy coming off and rolling off that way. That's just a copy of one of the old overhead that used to do this. This is a replication video. I pretty much talked about most of this, and you can play those if you want to see the animation. I think we're going to move on to other things. In this one, this is that single-stranded DNA that we're talking about. I just wanted the point of this slide is not to sit and memorize all this kind of thing. What's really interesting with viruses that one of the things I find interesting is how much information is packed into such a short area. Now we can see some genes here are fully in the genome. They're a big part of this single-stranded DNA. What's also interesting is that depending upon the way that we have transcription go on, we have overlapping genes that also show up in that single-stranded DNA, which I find to be very interesting, that you can have different genes within inside of genes on some of these things. Again, it's a conservation of space kind of thing. So look at for that. Don't go and worry about memorizing the different genes here and stuff. But think about the idea that even if a single-stranded DNA, you can get a lot of information packed in there, especially if you start thinking a little bit more creative, or selecting for a little bit more creative way of having genes that are overlapping. This we've kind of talked about. This is in the MS2 virus, just another mechanism. I want to show you a few different things. That we have the genetic map. You look at this. This is a really small piece of genetic material. Okay, and so this is an RNA phage. It's positive. So what happens? You have a minus strand synthesis that goes through then and resynthesizes the plus strand. It goes through both steps in this particular virus. Okay, at that point, this sense strand, some of it can be used to go into packaging of this. So this is this. Okay, so that continues to be churned out. Also, at some point, this then is made into the early proteins that are made that can come through and then help to transcribe the genes on this the original RNA also, together making a lot of proteins all at once and then this assembly. So you have this overlap of information going on that's all working by this chronological clock to make these viruses infect, do their thing, and then burst very rapidly. So that's the advantage of the plus single-strand RNA that I keep talking about, that is very effective. This slide is not in your book. I'm not even sure where I got it anymore, but I like talking about it as a thought question. That is implications for evolution. I don't know why there's archaeviruses. And I don't know why there's any rule that says that everything living has to have a virus that infects it. But I don't know, everything does, but a lot of things do. Okay, so in my mind, not that this is therapy 101, but perhaps in yours also, what is the evolutionary nature of viruses? Why did they evolve? I'm still not certain on that, but I do know they have evolutionary implications. So it takes a minute to read through this the way that it works. But what we have here is the type of phage, okay, and you kind of got to read this down to see what's going on. You have how it's being infected, and then what it's doing to Darwinian fitness. Okay, fitness in evolution is defined as what? It's not how many chin-ups things can do. What's biological fitness? Evolution, the creed of all us biologists. We know this. When we talk about Darwinian fitness, what is it? Survival, reproduction, giving viable, usually thought of as fitness. Good. Okay, so yeah. So in the temperate one, we have lysogeny. Okay, the lysogenic pathway, that one, the temperate phages we just talked about. Fitness is high. Not only the initial, but the chance to move genes across can also behind. If we have the lytic pathway, okay, and you have lysis, really there's not a lot of evolutionary implication of that. So there seems to be a benefit to having lysogenic viruses. So even if viruses started out this way, or it's an independent way that they do their mechanisms, this seems to have more of an implication for fitness, okay, especially through transduction, if you can move across genes that may be of benefit to the host. Okay, that's usually what happens here. To have latent bacteria, if you have a chronic infection, it's always in the back, or if it, I don't see that here, but if it goes into a latent type of phase, especially in animal viruses, that is an evolutionarily smart strategy, okay, if you play the probability game. Viruses aren't probably thinking about anything. That's the wrong way to think about it, but it's the way we often describe things. You know, chronic is a much better way to deal with hosts than having complete lysis or death. As people always say, well, if it's a really good virus, you know, it'll know how to keep its host alive. If, viruses that aren't so good at doing what they do kill off all their hosts. So there's different implications for evolution from viruses, independent of the question of how bacteria or how viruses evolved to begin with. Okay, I want to talk about viral diversity. I'll take a break. I gave a test yesterday, and this is a question of what would you do? I had to give this test, and someone's cell phone went off, which isn't unusual. I don't get mad about that anymore. It's like, well, it happens. Can you turn that off, please? And everyone reaches for their phones, especially when they turn stuff off. And there's this recorded message, and every 30 seconds it keeps saying something. The house is going to blow up, or I don't know. It's going to. And so, you didn't, you just kind of hit the dark button. Everybody turned everything off. And now at this point, I have students like angry at me. Fix it. It's like, okay. So everyone turns up everything, and low voice, okay, whatever it's saying. Your pants are on. I don't know what it's saying. So this continues to go on. They never could figure it. The students could not figure out how to turn this thing off. And so it was another not so good test experience for me. What would you have done? I mean, I didn't want to go frisking people's backpack. I didn't want to go and say, oh, you don't know how to turn off your phone. Well, you guys are as smart as me. You could all be professors. I didn't know what to do in the end, but you know the kicker, you know what it was? What do you think it was? It was a phone. So the tell at the end, we could all hear which backpack is going on, but now she feels bad because she's embarrassed. I'm embarrassed for her. So I wish I could turn your phone off. But she did. She's look, it's off, it's off. And, okay, she's going out of the room and she opens up her backpack and she says, my mom put her phone in my. She didn't know another phone was in there. It was kind of like one of these spy movies, So anyway, why did I tell you that story? I don't know. It was like one of those what you do, but I want to break it up a little bit before we get into viruses and death a little bit. So that was my little segue. I don't know. So we have viruses showing up all the time. Okay, I think one of the scariest things about being alive is that there's viruses around. And I think one of the most fascinating things about being alive is that we're still alive. The more I learn about this, and I think others, the more you learn, the more you'll agree. Like, oh my gosh, the immune system, that is just the cat's meow. I mean, without that, we would be gone. So let me show you some of these things, some of the things we can fix, some of the things we can't. We as society, as the world that gets concerned about these things, and we'll see what we think together. I think this is the flu last year. I tried to bring in a few flu updates. Okay, I'm not going to talk too much about plant viruses. And in fact, I was really happy to see someone on the list who's going to talk about the tobacco mosaic. That is the most famous one, very well studied. I'm sorry, I forgot whose name was on with that, but whoever's doing that, I'm looking forward to your report. You should be able to find a lot of stuff on there, as well as advances in it. So, you know, tobacco, I mean, it looked like dry scale. People said, well, you know, that's a virus, and then they started investigating it. Another one that interests me, though, that I find quite interesting is that as a person that studies lakes, I'm very interested in why algae blooms, and we've connected that to phosphorus and nitrogen in the 1960s. Eutrophication seems to be the word for everything in water these days. But that actually, you know, in the last decade or two, people say, well, you know, when those blooms go away suddenly, it's not just that they run on nutrients, it could be due to viruses. So there are some information on that. I just thought I'd throw that out there for some, you know, some people like medical stuff, some people like, you know, plant stuff. There's an algae thing that is very interesting, and you may find it to be interesting also. You do see this in a smattering still in the literature. People are very interested in that question. Okay. So let's do a little comparison here. In prokaryotes, the genome enters the cell. There's no compartmentalization. There are no compartments to really deal with. So in most cases, that's at least how we think about it. In eukaryotic cells, we may or may not have the entire nucleic capsid go in. Usually, the nucleic capsid goes in, the envelope stays at the surface, usually. The virus may make it to the nucleus. Now, in general, if the virus has an RNA genome, it tends to replicate in the cytoplasm. The machinery's there to do the stuff, right? I mean, why bother? I mean, that's where amino acids are synthesized. If it's DNA, it tends to migrate to the nucleus. Okay, either the nucleocapsid or the genome, but usually the nucleocapsid will just kind of keep moving. Okay, in general here, okay, transcripts must be exported to the cytoplasm. If it's DNA in the nucleus, it has to make it to the back out there. And this is really a simple diagram. I forget which textbook this is from, but it's what I taught some years ago, but I like it because it's simple. So now we see the DNA, this is an envelope DNA. It comes up, this is our, in this case, an animal cell. And we see that the envelope, generally with spikes or proteins with it, okay, those proteins and spikes can be of viral origin. They can sometimes be from the membrane of the cell. If they're from the membrane of the cell, they sometimes can be from the cytoplasmic membrane, sometimes from the nucleus membrane, nuclear membrane. Okay, so nucleocapsid comes in. In this case, it shows that we have an uncoating that looks like double-stranded DNA, makes its way into the nucleus. Okay, it's transcribed, replicated, so forth, and then can either go straight out. Sometimes in which case it's coated with one type of membrane. It can go out. It can have the nucleus membrane wrapped around it, and it can be a different type of coating for it. Okay, here they're just talking about the actual entering of the nucleocapsid directly. So there's different pathways, variants on the same theme, depending upon where in the Baltimore classification we're looking at. So I would say, if you always wonder, what are you going to ask me on the next test? Gosh, I'm not going to over the first one yet, second one yet, first one yet. Could you explain what's going on here would be the big thing. If I took out those captions, can you explain what's going on and the potential implications? Being coated with information around the envelope here is usually different than being coated with membrane information. Membrane information is really helpful a lot of times because the immune system looks at your cells and says, oh, that's you, and it doesn't eat you, okay, or send out defenses to you. It sees something else, it goes kill. You know, and it has different ways of doing that. This tends to give a little bit of radar, or escapes the radar with that kind of coating. Okay, animal viruses, they can look like a lot of different things. There's really, this is the thing about looking at shapes, not a lot of rhyme and reason. You know, that, double strand. So I mean, you know, good thing Baltimore came through and did what he did on those things. Here's RNA viruses. We recognize some of the names, you can get some relative sizes, but you know, it's kind of hard to make, to match the cards to the players given that. So, many more kinds of animal viruses than bacterial viruses. I think that might be more of a question that we're still trying to culture so many bacteria. How do you study viral infection if you can't study cultures? You know, to look at an isolated event of an infection and lysis of something you can't culture is very, very difficult. So I think this is more so a lay of where we're at in the field. I think, you know, and I think it's probably true that we do know more about that kind, but it doesn't mean that there's any less phages going on there, is my point. Okay. Okay, consequences of infection in animal cells. You can have a persistent infection, bang, infected, death. That's it. Latent infections, okay. You can have infection, it can either express or go latent and then express. So that's what latent is, shows up under times of stress or some kind of stimulus. Okay. Conversion. This is, viruses can create cancer. I guess that's no surprise to anyone in this room, but you can transform a cell. Okay, what is something I think we can all answer. What is a cancer? What is cancer? What is a cancerous cell? How can we describe that in terms of biology? Not what is a melanoma, what is, you know, the different types of cancer, but if we had to explain what a cancerous growth is, yeah.
Basically, it's an overgrowth of cells.
Yeah, it's undifferentiated, uncontrolled growth of cells. Okay, at one point, you know, in a way, you grew like a cancerous cell. I mean, we don't call you that. You're beautiful human beings and everything. But you know, at one point when you were, you know, a little zygote, you were dividing crazily, but you're differentiating all the way. So in a way, and it is very controlled. You know, so it's not just the speed of it. You know, and then you kind of stabilize, and then you get to be whatever height you are, whatever. So you guys got it. But you know, when you stop differentiating, you just start making growth, and it doesn't seem to be regulated. That's cancer, of course. Where is it located? What is it cutting off? What is it expressing? Those are all the things we have to consider when we consider cancer in its many forms. This just shows us the role of viruses in this. We have cell virus. You can just have attachment, penetration, fusion. What is the problem is if you have transformation and it gets into the genetic information and it causes a cancerous growth. Okay. If we just have lysis, it's not as big of a problem because you know, it's in the background. Okay. But it can have also a possible latent infection. It doesn't necessarily lead to cancer, but it can be certainly persistent in its growth. All right, polio. This is an example of a plus-strand RNA. So polio is really good at making itself and making it fast. Does polio still exist? Yes. Okay, but you know, talking about that generation, yes, I was born at the end of the 50s, way back. Yeah, I knew Fonzie and Richie and all the guys from Happy Days, and we all had molts and all that stuff. Anyway, but I just bring this up for a point. My mother explained to me that when she was in the hospital having my brother, who was born in 55, how worried she was about the polio virus. And indeed, she told the story how, you know, they shared rooms. You know, another woman was giving birth to a young son, and that baby got polio, and how happy she was that my mother or my brother didn't. And as much as I joke about my brother, I'm glad he didn't either. But in more or less a generation, there's things to worry about now, MRSA, tons of other stuff. We tend not to. We've kind of taken polio off the list many times in this country. It's more of a thing, well, you gotta watch out for it, but it's not one of those things like, boy, there's a good chance that's going to show up in my kid. You know, there's other things we worry about. So what is polio? 3 types of polio. It's among the smallest ones because it just has that little sense RNA. Boom, doesn't need much information. But I'll be talking about where these things go. It goes, so neurotropic, it goes to the nerves, okay. How can you get it? You can ingest it. You can live in tonsils. Well, a lot of things can live in your tonsils. I'm from that age where they locked out everybody's tonsils. I remember I got ice cream for a week. That was it for me. How many of you guys still have your tonsils? They don't take them out anymore, right? I mean, sometimes. Yeah, if it would have been my class, my graduating class, everyone said, no one would raise their hand. They just said, oh, take out the tonsil. So anyway, stuff could be in there, but there's a lot of benefits of tonsils too. Okay, what do you notice? Well, you're going to see this in all these diseases. Your body tries to get rid of it, and it makes you feel lousy. Okay, that can probably be said for most stuff as we go. But here's the key thing. Okay, this virus makes its way in the genetic tract, gets into the blood. Okay, it's not that hard for a virus to do that. Okay, you have absorbent areas in your body, the GI tract being one of them, viruses being really small. Okay, now in the blood, not yet a real problem. Okay, it's when it passes the blood barrier into the meninges of your body, then you have a problem. What are meninges?
In your brain, like the membranes are in your brain.
Yeah, they are, okay. And why is that a problem? Because then that's a great place to incubate and to infect and to cause havoc, and that's what happens. So basically, you're losing control of part of your nervous system, not voluntarily, but through infection. Paralysis of the arms and legs, this is the most common thing. Sometimes, okay, a lot of people were born with deformed limbs, okay, or develop that, but during the time that you're developing in your life. If it's severe brain swelling, yes, you have an infection perhaps in your brain, and that can cause death, both from brain swelling, but also you can have loss of your diaphragm. If your diaphragm isn't working, you can't breathe, you suffocate. Yeah, it's up there too. So, well, it was interesting. People were very worried about this at one point. I mean, like I say, now it's on the radar, but it's not the same thing. Jonas Salk looked at this and started to think about virology in a different way, and Albert Sabin looked at it another way. Salk said, what if we made some of this, you know about cowpox and Jenner, and they give the kid cowpox and the kid didn't die. You guys know that story? Virology? Anyone know about Jenner and smallpox? Okay, virology. Jenner was a guy in the 1800s, and people were dying from pox all the time. Okay, so, and as I remember the story, it's like one of the servants' kids that he had, I guess, servants. And he was just a compassionate person. He didn't want to see people dying, and this child was infected. He goes out to the barn and gets some cowpox off the cow. I don't know, I guess he just had cowpox thing on. So he grabs some of this stuff and he gives it to the child. The child doesn't die from the pox disease, smallpox. And so this is the first vaccination. Okay, so this is on the books. People know about this in the general sense. I want to tell you that story so I can tell you this story. So Salk is around. He says, okay, well, we can pull the Jenner thing. What if we give rabies viruses to people, but we don't want to give them a virus. We want to kill people. What if we alter it? Okay, and you see this to this day in altering different chemicals and viruses. They use formaldehyde. Now, you should know the answer to this. You've dealt with formaldehyde frogs, you preserve stuff from formaldehyde. Why did Salk choose formaldehyde to alter the outside of viruses? What does formaldehyde, so the more direct question is, what does formaldehyde do to proteins? It cross-links them, that's why it preserves it. I mean, you ever notice that have you ever noticed that if you have a specimen, I don't know, anything, you have a cat, you know, and your cat at home, wherever you see a cat or dog, your muscles are nice, like your muscles and stuff. You put anything in formaldehyde, it kind of turns into erasers, right? Okay, that's cross-linking of proteins. So, Salk uses formaldehyde, and where's the protein in the viruses? Everyone knows this answer?
In the capsule?
The capsid. So it makes sense, okay? We hope that you can preserve, you can cross-link it, they won't open up and kill everybody. That works really good. Sabin says, well, let's be a little more certain, let's mess with the nucleic acids. But anyway, they're able to attenuate the virus and to make a vaccine that starts to knock out polio. We used to get it on sugar cubes. I don't know how you guys get it now, when they give it to you or whatever. You're looking at me. I'm sorry. Thank you for your attention. You're a very polite and gracious class. We'll see a lot of you.